Hello, 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 hello. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, so cell division, it is very important process in all living organisms. During the division of a cell, DNA replication, cell growth also takes place. See, a cell during its division into two daughter cells, a small cell will grow into a larger cell, and then this larger cell will divide into two daughter cells, right? Small cell grows into a larger cell because the cytoplasmic content present here will also increase, as well as DNA present will also double. So during the cell division of a cell, DNA replication and cell growth also takes place. All these process, that is, cell will divide, DNA replication will divide, and cell growth. See, first cell growth takes place, increase in cytoplasmic content. Then DNA replication takes place. Then after, cell will divide into two daughter cells. So all these processes, what cell division, DNA replication, cell growth, have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. So all these should occur in a coordinated way. The sequence of events by which a cell duplicates its genome, synthesis of see all the sequence of events, what all the sequence of events occurring, cell growth taking place, DNA replication taking place, and finally cell division taking place, right? So all the sequence of events by which a cell, a single cell, duplicates its genome, synthesizes the other constituents of the cell, that is cytoplasmic constituents, and eventually divides into two daughter cells, is termed as cell cycle. At all the sequence of events that occur during the cell which is divided into two daughter cells, that is known as cell cycle. Although cell growth in terms of cytoplasmic, we'll say cell growth means cytoplasmic content is increasing. If suppose we will say this is a cell growth, means its cytoplasmic content is increasing. What all the proteins, phosphorus, sugar, nitrogen that are required for DNA synthesis, DNA replication, all these will be present in the cytoplasm, right? All these proteins, phosphorus, sulfur, nitrogen, sugar, all these will be synthesized in the cytoplasm, and they will be ready to be used by the cell to synthesize its new DNA. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage of the cell cycle. Cell growth, it is a continuous process. There is no particular stage. इसी स्टेज में ही सेल ग्रोथ टेक प्लेस, ऐसा नहीं बोल सकते. Any phase of the cell growth will occur continuously, but DNA replication occurs only in a particular phase, that is S phase. Only in a short particular phase, that is S phase. So DNA synthesis only occurs during one specific stage of the cell cycle. The replicated chromosomes, that is DNA, are distributed to the daughter nuclei by a complex series of events during cell division. So this replicated DNA, they are distributed to daughter DNA. This DNA replicated na, new strand will be synthesized by the daughter. See, single strand from each parent Each strand from parent goes to two daughter cells, and a new strand will be synthesized newly by the daughter cell. Replicative momentum distributed to daughter nuclei by complex series of events during cell division. These events are themselves under genetic control. So all these events, not all the phases G1S, G2M, all these will be under control of gene DNA. So we will come to the phases of the cell cycle. A typical eukaryotic cell is illustrated by a human cells in culture. So we will discuss about eukaryotic cell. These cells divide once approximately every 24 hours. So for every 24 hours cells will divide. However, the duration of the cell cycle vary from organism to organism and also from cell type to cell type. See, cell divides in 24 hours, right? But in this 24-hour duration, there are different phases of the cell cycle, and each phase has different timings. And those timings will be also species specific time limit for each phase of cell cycle. Duration of the cell cycle can vary. So there comes a bit like the cell cycle duration varies from organism to organism or from species to species. Yes, the duration will vary from organism to organism and cell type also, cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. Yeast, it undergoes cell cycle in 90 minutes. But eukaryotic cell, our human cells, they undergo 24-hour cycle. Right? The cell cycle is divided into two phases. We will come to phases of the cell cycle. Right? Now, cell cycle is divided into two basic phases, that is interphase and M phase. M phase is nothing but mitosis phase. So G1, S and G2 phase, all this is interphase. Right? This is M phase, mitotic phase. The M phase represents a phase when the actual cell division or mitosis occurs. This is the phase where actual single cell divides into two daughter cells. Actual cell division or mitosis occurs, and interphase represents a phase between two successive mitotic phases. See, M phase, interphase, M phase. Interphase occurs between two successive mitotic divisions. So first mitosis, then interphase, then mitosis, and this continues like this. Then interphase, then mitosis. So interphase is a phase between two successive mitotic divisions. Phase between two successive phases. It is significant to note that in the 24-hour average duration of cell cycle of a human cell division proper lasts only for about an hour. See, during this entire cell cycle, what are we telling? G1, S, G2 and M phases. These phases timings will vary, and cell division occurs in mitotic phase only 1 hour. Within 1 hour cell division occurs, and the remaining G1, S and G2 phases takes long duration. These three phases will take long time when compared to M phase. See, M phase takes 1 hour, G1 11 hours, S phase 8 hours, G2 phase 4 hours. Okay, this is the time period of a human cell. The interphase lasts more than 95% of the duration of the cell cycle. See, 11 plus 8, 19, 23 hours. 23 hours of the cell cycle occurs because entire time period will be consumed in interphase only. Matlab the cell interphase is the phase where the cell will make all its constituents ready. So protein, phosphorus, etc. all will be ready during interphase. DNA replication also occurs during interphase. That is S phase of it. S phase of interphase. Right. So it takes longer time. So what all the constituents after synthesizing they enter into M phase and immediately cell will divide within 1 hour. Interphase lasts more than 95% of the duration of the cell cycle. M phase starts with the nuclear division corresponding to the separation of daughter chromosomes. See, a cell if it wants to divide, it undergoes first nuclear division, that is karyokinesis. Then it undergoes cytokinesis, that is cell division. Karyokinesis means nuclear division, cytokinesis means cell division. First nucleus divides. See, in a cell first nucleus divides, then after, see, I will tell you. In a cell first nucleus divides into two, then only we will get two daughter cells with two nuclei. Okay, first karyokinesis takes place, then only cytokinesis takes place. The interphase, though called resting phase, is the time during which the cell prepares for division by undergoing both cell growth and DNA replication in an orderly manner. See, I told you na, interphase, though it is called a resting phase. Resting phase for what? Resting phase for synthesizing all the constituents required for cell division. See, during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. Interphase is again divided into three phases. See, I told you na, interphase G1, S and G2 phase. G1 phase, gap 1 phase. S phase is known as synthesis phase. G2 phase is known as gap 2 phase. Okay. G1 phase corresponds to the interval between mitosis. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. See, G1 S, G2 N. I'll draw here. Okay, I'll draw here. M phase G1 S G2. Okay, G1, what he is saying, G1 corresponds to the interval between mitosis and initiation of DNA. Initiation of DNA replication occurs in S phase. So G1 phase is the phase between mitosis and initiation of DNA replication. G1 phase, it is a phase between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. Here it will be metabolically active. I told you na, the cytoplasmic content will grow. So what all the requirements, nitrogen, phosphorus, sugar, etc. will be synthesized in G1 phase, but replication will not occur in G1 phase, but does not replicate its DNA in G1 phase. Okay, synthesis or S phase marks the period during which DNA synthesis or replication takes place. S phase is a phase where DNA replication takes place. During this time, the amount of DNA per cell doubles. So S phase replication, what is replication? A single strand will be divided into two daughter strands, right? S phase replication, during this time, amount of DNA per cell doubles. Amount of DNA per cell doubles. See, this is the actual chromosome, right? After DNA replication, the chromosome will look like this. Why? This is the chromatin present in the chromosome, in the condenser state of chromosome, right? After S phase, the chromosome will be like this. Chromosome number is same. Here there is one chromosome, here also there is one chromosome. If this is one chromosome with one arm, arm means chromatid, one chromosome with one chromatid. Here one chromosome with two arms, that is two chromatids. Why two chromatids have taken place? Because DNA replication has taken place, right? The initial amount of DNA is denoted as 2C, then it increases to 4C. See, if the DNA is 2C, then it will increase to 4C. If the DNA is 1C, it will increase to 2C, right? However, there is no increase in the chromosome number. See, no chromosome number will be increased after S phase. One chromosome initial and final also one chromosome only. No increase in the number of chromosome number. Only chromatid arm will increase because of replication of DNA. That is only chromatid. We can't say it as two chromosomes. There is a single chromosome with two chromatids. However, there is an increase in the chromosome number. There is no increase in the chromosome number. If the cell had diploid or 2n number of chromosomes at G1, even after the S phase, the number of chromosomes remains the same. See, if this is G1, suppose this is a cell having a diploid number of chromosomes, even after the S phase, it will be having diploid number of chromosomes only. See, I'll draw it neatly. This is a diploid number of chromosome. Same number of chromosome, diploid and diploid even after S phase. What is the difference? Only an extra arm has added. Extra arm has added because the DNA has replicated, right? Chromosome number same. See, if cell has diploid number of chromosomes in G1, even after S phase, the number of chromosomes remains the same. In animal cells, during S phase, DNA replication begins in the nucleus and central duplication in the cytoplasm. See, I told you now, in cytoplasm what happens? All the constituents required for cell division occurs, right? DNA replication begins in the nucleus. We all know that in S phase DNA replication, this is the nucleus containing DNA. DNA is present in the chromosome, right? DNA replication occurs in the S phase of nucleus of the cell, and in cytoplasm what happens? Centriole. See, wait. In cytoplasm, centriole duplicates in the cytoplasm. Here centriole duplicates in the cytoplasm. During G2 phase, proteins are synthesized in the preparation of mitosis while the cell growth continues. In G2 phase what happens? Proteins will be synthesized. Proteins, what proteins? Microtubular proteins. Why microtubules are required? Because during cell division, microtubules are needed to drag the chromosomes apart. We need microtubules. These microtubules will be synthesized during G2 phase. Okay, in G1 phase, phosphorus, nitrogen, sugar, etc. will be synthesized. S phase, replication takes place, and in G2 phase, microtubule, etc. proteins will be synthesized. So finally the cell is ready for cell division. Okay. Some cells in the adult animals do not appear to exhibit division. Heart cells. So some cells like heart cells, they will not divide, and many other cells divide only occasionally as needed to replace the cells that have been lost because of, see, some heart cells, there are some exceptions. Heart cells will not divide continuously, they will divide for a limited period, right? After that they will enter into a Q0 stage, that is quiescent stage. This is a quiescent stage, Q0 stage. This is an inactive stage. The stage is inactive, but cells in this remains active. Cells in this stage remains active, but the stage, this quiescent stage is inactive. Means active cells will enter into quiescent stage, and only if it becomes necessary, that is during injury, they will come out and they will replace the injured cells. See, as needed to replace that have been lost during injury or cell death, they will only divide, some cells divide occasionally. These cells that do not divide further exit G1 phase and enter into an active stage called quiescent stage. These cells, cells that is no longer required for the body, they are active, cells are active, but they are no longer required for the body, they no need of extra cells, so they will enter from G1 into Q0 stage, quiescent stage, in order to save the cells. That is, whenever required for, whenever if injury happened, this cells from Q0 will again enter into the injured part and they will replace the cells. Cells in this stage remains metabolically active but no longer proliferate unless and until called on to do so, depending on the requirement of the organism. See, they will no longer proliferate, they will be active, they will be metabolically active but no longer proliferate. In animals, mitotic division is only seen in the diploid somatic cells. See, mitosis occurs in diploid cells or somatic cells or body cells, we can say body cells. Mitosis occurs in somatic cells, matlab other than germ cells. Other than germ cells, mitosis occurs in somatic cells, right? Germ cells, what are germ cells? Like sperm, mother cell, ovary, right? Except in these cells, remaining in all the cells, mitosis occurs. In these cells, meiosis occurs. Germ cells, meiosis occurs. In somatic cells, mitosis occurs. See, mitosis is only seen in the diploid cells. However, there are few exceptions. Mitosis, if it occurs in, mitosis, if it occurs in diploid cell, it finally gives rise to diploid progeny. If meiosis occurs in a diploid cell, it finally gives rise to haploid progeny. This occurs in germ cells because here spermatogonial mother cell will give rise to sperms finally, and ovarian mother cell will give rise to ova, right? This occurs in meiosis, germ cells. Mitosis occurs in somatic cells. So here both daughter and parent has same identical number of chromosomes. It is hence mitosis is known as equational division. Meiosis is known as reductional division. Why? Because here chromosome number is getting reduced to half, right? There are few exceptions where haploid cells divide by mitosis. Mitosis also occurs in some haploid cells. Example, in male honeybees. In male honeybees, mitosis occurs in haploid cells, and also some fungal plants also, some fungal plants, some fungal cells undergoes mitosis. Against this, the plants can show mitotic divisions in both haploid and diploid cells. So, alternation of generations in plants. So, he is asking the question. Okay, we will discuss this later on. Okay.